just let me. Okay, so we are now in YouTube. Okay, so thank you. Good afternoon to everybody. Thank you very much for being here. Today, it's a real pleasure for me to introduce Dr. Aaron Bogan, who is going to be presenting today's seminar. Dr. Aaron Bogan is a young scientist from Uppsala University in Sweden, who is interested in understanding and has made important contributions to our understanding of the evolution and organization of the genome in eukaryotic organisms, most notably in fungi. Dr. Aaron Bogan obtained his bachelor's degree from the McMaster University in Hamilton in Canada, where he also performed his PhD studies in biology. And then in 2016, he moved to Sweden, to the Uppsala University, where he joined the lab of Hanna Johannesson to do his postdoc studying meiotic driving in different fungi, including species of Neurospora and Podospora. In 2020, he started his own research group in the same university, in the Department of Organismal Biology, in the section of Systematic Biology, in a university with a large tradition in systematics, you know, the University of Carl Linnaeus. So it's an amazing place to be studying systematics. And he has already mentored numerous students, both grad and undergrad students. He has performed research exchanges and stay in different places, including the University of Innsbruck in Austria and the Pacaya-Samiria National Park in Peru. And he has been recipient for multiple awards, including numerous grants and fellowships from Sweden, Canada, etc. And despite his young age, Aaron has made really fascinating discoveries understanding the evolution of genomes, including the identification of mobile autonomous elements and their impact in genome shaping and inheritance. And his research has been published in over 50 publications in very renowned journals. And currently he has expanded his research focus into understanding how these genomic remodeling events participate in defining genomic traits involved in promoting virulence in particularly in fungi. So without further ado, please let me welcome Dr. Aaron Bogan. Thank you very much, Aaron, for accepting the invitations. Please.
Thank you, Leonardo, both for that great introduction and for inviting me to do this seminar. And thank you as well, everybody who came to listen to me talk about this. Yeah, it's really great for me to have the opportunity to do this. So yes, as Leonardo mentioned, I'll be focusing this talk on some of my recent work on horizontal gene transfer in fungi and a new sort of class of elements that we've decided to name starships. And so, as many people might have heard, there was a recent sort of study that came out saying that about 1 million deaths in 2019 could be attributed to antibiotic resistance in bacteria. So it's a very large concern, one of the greatest challenges that we're going to be facing today in infectious biology. And one of the reasons it's such an issue is because, of course, bacteria can share genes horizontally. So if we consider sort of most transmission we think of goes from sort of parent to offspring, and that can either be clonal, or if you have sexual reproduction, then that would also be vertical transmission as well. But there are many systems, particularly in bacteria, where individuals can share genes horizontally. So conjugation, often considered sort of bacterial sex, where they can share genes that way. It could be viral mediated, also called transmission, or through environmental uptake, which is often what we consider to be transformation of DNA. And so these are all very important mechanisms that we know that prokaryotes use in order to sort of acquire and adapt, acquire genes and adapt to novel environments or stressful environments. But my focus is on fungi, and if bacteria are sort of the biggest threat maybe for infectious biology, fungi are the most important in terms of food security and also overall ecosystem health because of the way that they interact with various plant species. And this can be sort of directly as pathogens or as symbiotes and endosymbiotes, or it can be more indirect as sort of important for nutrient cycling. And they form sort of the basis of some ecosystems in that respect as well. And what may not be so well known outside of the field of mycology is that fungi actually do have lots of examples of horizontal gene transfer, both among fungi, like between unrelated fungi, as well as various examples where important genes have been shared from fungi to arthropods or to plants, and also potentially acquired from bacteria. And I'll just give a quick sort of list of some interesting ones, and also just sort of highlight that these aren't random individual genes sort of that seem to be moving, but actually large, intact, functional gene clusters. And so the ACE1 gene cluster is important. That's been moved from Megaporthus, so from rice blast, to a species of Aspergillus. That's very important for causing disease in rice. Another one that's very beneficial to humans is that actually wine yeast managed to acquire sort of a 7-gene cassette from a different species of yeast, from Zygosaccharomyces. And then another one that's probably the largest identified is a 23-gene gene cluster for a mycotoxin that had moved from Aspergillus to Podospora. But we actually have, up until now, had no idea how any of this horizontal transfer is mediated. We could sort of see the signals of this in the genome at a genome-wide scale, but how this is actually occurring has been unknown. And that's sort of, I think that we've discovered how this is happening, and I'll try and walk you through sort of the whole study from the beginning, the whole story from the beginning and end of how we found this. And the story begins with what is the greatest model organism. This is Podospora anserina. Hopefully you've heard Leonardo talk about this and already convinced you. If you don't work on this organism yet, you should. I have studied many different things, and time again, the answers have been found in Podospora. So I highly recommend working on it. And so where I sort of first came onto the story that eventually led us to these finding answers was working on meiotic drive. And meiotic drive can be thought of as these cheating genes. There's certain genes or loci that are able to sort of manipulate normal meiosis in order to bias their transmission to the progeny. And so here you see sort of a normal rosette of Podospora. So in Podospora, they produce 4 spores per round of meiosis, and they package them together. So this is one of the reasons it's such a great system, is you can sort of just visualize the output of meiosis. But if you cross strains that differ in these spore-killing genes called SPOCs, what you'll start to see are asci, so outputs of meiosis where you have half the number of progeny. And what's actually gone on in this case is that these 2 spores that you see living here killed their sibling spores. And that's due to these meiotic drive genes, these SPOC genes. And we knew about this one gene when we started studying this, but we knew that there was a lot more spore-killing phenotypes out in the population. So we wanted to understand this a bit better at the population level. And so we sequenced a whole bunch of natural strains of Podospora that showed variation in these spore-killing phenotypes. And one of the great things about working in Podospora is that we have these wild collections, and they are very, very, very similar. They're more than 99.9% similar at the genomic level. So when you have genes of interest, you can really narrow down to what it is, because there's otherwise very few differences. And so here I'm showing an alignment of one of the chromosomes, chromosome 5, from 3 different strains. And the blue are the completely synthetic regions. And you can see that, again, these are very, very similar. But you see sort of some messiness. That's just a bit of transposon movement. And then other than that, you have this big block here that is sort of moving to different positions or completely absent. And that really stood out to us, obviously. And when we looked at what this block contained, it contained our SPOC genes, so the spore-killing genes, at least 2 of them. And although there were some differences among both the SPOC gene content of these blocks and their position, they were otherwise the same. So here's sort of an alignment of many different sort of iterations or versions of this block from different strains. And again, the blue is showing synthetic regions. The red are some inversions. And the yellow here are some internal duplications, but otherwise they basically have the exact same gene content. And so we were able to then explain all of this different mitoid-derived phenotypes, all these different spore-killing phenotypes, by realizing that there's actually 3 spore-killing genes in this species, and 2 of them jump around inside this large block. And you get so many different phenotypes out in the wild because it seems like this whole block is moving. And that was really interesting because it's quite large. It's over 200 kb. This is much larger than a regular transposon in eukaryotes that is maybe 10 kb. So how this was occurring, we wanted to figure out how this was occurring, how this was moving around the genome. And I'll sort of spoil the answer first. It turned out that it was moving because it was a giant transposon. And we ended up naming it Enterprise because it carries the Spock genes, of course. And for those of you who aren't used to thinking about transposable elements, I'll run through just a very, very quick sort of primer on how they typically work. So there's usually 2 flavors that we talk about, or classes. The first are the retrotransposons that use an RNA intermediate in order to copy and paste themselves around the genome. And these sort of max out typically around 15 kb. They have a few different orders. The LTR type is sort of the most usual one that's talked about, that have these sort of structural features on either end. The other type are the DNA transposons, and these are typically much smaller because they only contain 1 gene and integrase. And these typically move themselves around through a cut and paste mechanism with no RNA intermediates. And there's a few orders of this as well, and one that I want to highlight in particular are called kryptons, because they're very understudied. They're widespread across eukaryotes, but there hasn't been a lot of research on them. And they're a bit difficult to study because they don't have any of these structural features that some other transposons have. But the important part is that instead of the normal DNA integrase, they actually use a tyrosine recombinase and presumably transfer by inducing holiday junctions in DNA. And this will become relevant in a little bit. But to get back to the story of the Spock block, this large region in Perospora and Serena that seemed to be jumping around, we were able to sort of demonstrate that it is moving through a transposition mechanism. And I'll try and quickly walk through how we figured that out. The first was by analyzing the insertion site. So because we had a lot of high quality genomes, we could see what the edges of this block looked like in strains with it versus strains without it. And it was always very clean insertions of this just dropped right in. And additionally, we would see this little motif repeated, the 6 base pair motif on both ends, which could be a target site duplication. It's sort of a hallmark of many transposons, especially DNA transposons. We also saw that the target was always the same. So not only was this duplicated, it always targeted AGGTAG, which is also another hallmark of some transposons. But it was, these blocks are very big. So we knew that the spore killing genes were in there. But if we wanted to understand how this could possibly be transposing, how it could be jumping around the genome, we wanted to know what genes within this block could be mobilizing it. And so I'm just showing a circles plot that this ended up being a fairly difficult question to ask. So here's sort of the genome of one strain, with orange showing the gene density and red showing repeat content. And if you look where the Spock block is, there's not a lot of genes actually annotated in it. And that's partly because it seems to be full of pseudogenized genes for the most part, and a little bit of TEs, but not so much. And this is just that block blown up over here. And so we sort of looked at where we had the best gene models and also the most conservation among the different versions of the block. Sorry, and here's a better zoom in of this. And when we did that, we noticed sort of the very first gene that stood out as being of interest. It was always there. We had good gene models for it, etc. And we hypothesized that this is the gene responsible for actually moving this large block. And so we named it Kirk for killing related krypton, because we think it's actually related to these krypton type transposons that I introduced a little bit before, that's actually using a tyrosine recombinase mechanism to move this whole block in and around the genome. It previously had a domain of unknown function, this DUF3435, but we did some bioinformatic analyses, and we think that it really is a tyrosine recombinase. And furthermore, this is sort of a completely novel tyrosine recombinase, which is part of the reason it was just labeled as domain of unknown function before. So here's sort of a massive tree of all known tyrosine recombinases, including a lot of prokaryotic ones, and then also sort of in the more rainbow colors, ones from known transposons in eukaryotes. And it's a very messy tree, but just to show that in sort of this greenish color are these new group of tyrosine recombinases that we discovered. So they're not related to anything that we previously knew about. And for us, what was really the smoking gun that this really was a transposon was being able to find it in another species. So we sort of looked for the same hallmarks of the transposition that we were seeing in Prospera serena and searched a large database of fungi, and we found it in this other thing called Melanconium sp, where it was present actually in 4 copies with the exact same sort of hallmarks of transposition that we were seeing in Prospera serena. So that really settled it for us. But of interest, this one didn't have any of those spore-killing genes. There were no spocks here. So that led us to sort of come up with this hypothesis that these new tyrosine recombinases sort of represented a new type of transposable element in fungi, and that they could either just exist perhaps on their own with other genes, or they could grow to larger sizes and incorporate a lot of host genes or other exogenous genes from that. So we thought that what we were seeing in Prospera serena, the Spock block, was sort of a unique combination of this transposon plus these meiotic drive genes. And so that sort of sums up the first part of the story. And just to quickly summarize, so the Spock block was we called it a hyperselfish genetic element because it had both the Spocks for meiotic drive and Kirk for transposition. But we saw that this seemed to be a wider phenomenon in fungi, and in particular, we found this enterprise transposon in other species. And then we had also shown that this was probably detrimental because we did some fitness assays as well, which I didn't talk about. And now the story is going to jump from our nice model species of Prospera serena to a very interesting plant pathogen called Macrophomina phaseolina. And this is not closely related to Prospera at all. It's a totally different class of fungi. And it's relevant because it affects as many as 500 different plant species, including many crops. Basically second most prolific plant pathogen after Fusarium oxysporum. And my collaborators on this, they had really nice samples. They collected over 300 strains from Paraguay and Ohio in the US. And they had generated high quality genomes, 6 from Ohio and 6 from Paraguay. So we started off with a really nice data set. They had done some just straight up phylogenetics and genome assembly and things like that, and had already started to see some interesting variations. So here you can see how large the genomes are, and they vary from below 50 megabases to over 60. So there's quite a large amount of variation in genome content, even though some are basically almost clonal. You see very little phylogenetic difference among orthologs. And so they dug into that a bit more. And again, they found that among orthologs, these are all very, very similar strains, more than 99% similar. But among gene content, they could vary drastically. And so for gene content, some varied by as much as 70% differences. And you can describe this as a pangenome, so the collection of all genes among all the strains in the species. And it ended up being quite large. So again, Macrophomina is here in red, and it seems to have the second largest pangenome, again, behind Fusarium oxysporum, who's sort of a more model plant pathogen that's well studied versus this sort of not as well studied Macrophomina. But we clearly have a lot going on in this genome. And when we sort of categorize where those indels are, like where are all of those genes that vary between the strains, they are compartmentalized into sort of hotspots. And it turns out that they're mostly found in large hotspots. larger than 10 kb. And so this started ringing bells in my ears of, this looks exactly the same as Enterprise. So we probably have something similar going on. And I started looking and I found what became Voyager. So again, we ended up finding a large transposon that seemed to be moving around these genomes. There was very little in common with Enterprise, though, other than that it still had this putative tyrosine recombinase. And it had some of its own unique features that we were really interested in. So for instance, it seems like its target is actually the 5S rRNA gene, which should be bad because if you lose your 5S rRNA genes, you can't build your ribosomes and you can't make proteins. So that really stood out. And so we sort of quantified how well these strains can grow versus how many copies of this new TE Voyager they have. And so in the population we had, or in our sample, we had strains with between 0 copies of Voyager and up to 5. And we ended up seeing a trade-off. So the more copies of Voyager they had, the more infectious they were on plants, on soybean, in this case that we were surveying. But when we grew them on sort of rich media in the lab at a variety of temperatures, it seemed like the more copies you had, the worse you grew. So putatively, having this large TE is incurring a trade-off between how good a pathogen you are, but since Macrophomina can grow without a plant host, it can grow in the soil, it would then be worse at that if it got these elements. And so we looked into Voyager to see what could be driving this, and we found a menosyl transferase, which is actually being characterized from other plant pathogens as being important for virulence. And so it seems like by having more copies of Voyager, you're increasing your copies of this and potentially becoming a more severe pathogen. And yeah, and so with that, we got interested in the rest of the gene content in Voyager as well. And so we did, we took all the gene content of Voyager and we took a large library of 1,500 fungal genomes, and we sort of ran an analysis to see how many other Voyager-like elements might be out there. And we found a lot. And to our surprise, there was other conserved genome content. So basically down here are all the hits to just that putative tyrosine recombinase, what we think is the captain. And then in this subtree, these are all the ones that had a larger auxiliary gene content that was shared. So they all sort of by definition have the putative tyrosine recombinase. And then many also had this other domain of unknown function gene, as well as a putative iron transporter, some NOD-like receptors, and also a ptyalin-like phosphatase. And this sort of seemed to make up a core unit of these elements. And that was all done automatically. And so we then went and dug into these genomes manually to see if we could annotate these in the same way that we had with Voyager and Enterprise previously, with those sort of characteristic transposon-like motifs. And we pulled out over 24, so over 20 that we managed to find. And since then, I've found many more. I think I'm up to almost 100. So these are very widespread all across filamentous fungi. And so we decided that they needed a name. And given the name of the first couple elements, we decided to call them Starships. And they are mobilizing a large amount of genes and genetic material throughout filamentous fungi. We think it's tyrosine recombinase mediated transfer. Other than this tyrosine recombinase though, the other genes, which we call cargo, are very different, basically entirely different between elements, other than this sort of set of auxiliary genes that we found. And they're present across Mycotina, which are sort of typically the filamentous fungi, including fungi with many different niches, plant and animal pathogens, lichens, mycorrhizal fungi, you name it, we find these things there. And so now we're sort of trying to dig in and figure out, what are these really? We can see these, are they just sort of selfish giant transposons, or maybe there's something more? And so now we're going to go back to thinking about bacteria and prokaryotes that I mentioned at the beginning of the talk. So when prokaryotes trade genetic information, you're probably used to thinking of this sort of complex pilus, so this type 5 secretion system, where they actively sort of share plasmids, or things called integrative and conjugative elements, ICEs. And this is sort of, the ICEs themselves usually encode for all the machinery to actually activate this and send the genetic material from one strain to an unrelated strain. But that's not actually how all prokaryotes do this. Actinomyces, so things like streptomyces, they actually trade their ICEs through hyphal fusion. So, right, so actinomyces are these filamentous bacteria, and they can actually do hyphal fusion. And it turns out that the ICEs themselves encode genes that will promote that fusion and then promote the transfer of the element between unrelated strands. And so, of course, fungi have hyphae, and they can also do hyphal fusion. And actually, we also have cases where we do see genetic material moving between individuals, including plasmids, which has actually been done in Prosper and Serena as well. And so if we look at some of the important things that these actinomyces ICEs have and what the Starships have, we see a lot of similarities. So they both tend to target or can target the rRNA genes in the genome. They're usually tyrosine recombinase mediated. They tend to have these helper proteins to stabilize sort of the circular intermediate as they transfer from one to the other. And we think that might be the role of some of these sort of auxiliary genes that we discovered that are this other domain of unknown function. Another interesting thing is that they typically transfer among more closely related individuals. So if you're used to thinking about bacteria just doing their conjugation with anyone, that's not really true for the AICs. It's usually among sort of conspecifics or things in the same genera. And that seems to be the same signal we're seeing in the Starships. And then similarly, they have a huge diversity of cargo genes. They carry toxin antidote genes, which actually could be what the main purpose of the Spock genes are, getting back to that first question. And then, of course, they do conjugation via hyphal fusion. So that led us to believe that these fungal Starships are eukaryotic analogs of prokaryotic ICEs, and specifically these actinomyces versions. And so we have to show that now is the challenge. And I'll now jump over talking about another element named Hephaestus. And so somewhat coincidentally, at the exact same time we dropped our preprint of Enterprise, another group working in Australia dropped their preprint of Hephaestus. These came out within 2 weeks of each other. And they'd also seen this large chunk of DNA moving around between strains of this time a fungus called Pacillomyces. It's an important contaminant of crop. If you have a lot of grain in a silo, this will make it go bad. And Hephaestus was interesting to them because in yellow here are all genes for heavy metal resistance. So this, they were able to show at least 5 heavy metals, including zinc and cadmium and arsenic, that this basically massive gene cassette codes for tolerance to or resistance to. But they didn't know how it was moving around. They didn't have a putative mechanism. But when I looked at what the very first gene was in Hephaestus, it had the same domain of unknown function. This is our putative tyrosine recombinase. So this really hit it off that we're looking at the same phenomenon, in fact. And so I started talking with them, and Andrew Urquhart, who was the first author on this paper, has now followed up on this and done some really great work digging into the mechanism that's going on here. And I'll present some of his very new data now. So up until now, we've sort of been assuming that these things are active and moving in the genome because when we compare very closely related strains, we see them in different locations and we see them sort of as clean insertions. But we haven't seen them move. That's sort of the gold standard. You want to watch these things jump. And so Andrew set up some really clever experiments in order to do this. And so the first thing he did was to take sort of the native genome, the native strain that had Hephaestus, and put an antibiotic-resistant gene on either side of it, essentially. So the main part of the gene was on one side of Hephaestus, but its promoter was on the other. So normally it doesn't function. If you just put that strain on media with the antibiotic, it won't grow. But if Hephaestus excises itself and moves, then that brings both ends together, and it's now resistant to that antibiotic. And so he designed this, he set it up, he implemented it, and he saw movement. So yes, Hephaestus is mobile. So basically, here's sort of the control where now that Hephaestus is left, all of these strains are hygromycin resistant. He also, because Hephaestus carries genes for all these heavy metal tolerance, he could screen to see if Hephaestus made it back into the genome or not by growing these strains as well on cadmium or zinc. And as you can see, actually, although it excites itself in all these cases, it doesn't always make it back into the genome. So it's not a perfect mechanism, it seems like, which we're also pretty interested in. He then wanted to dig into this a bit more and figure out if we were right, that it is this first putative tyrosine recombinase that's moving these transposons, that's moving Hephaestus. So again, there's many genes in here, actually. So we wanted to narrow down, is it actually this first gene? Is this the captain that's moving this? So he designed what he calls a mini Hephaestus. And he did this on a plasmid with basically the same design, with sort of the split antibiotic resistance cassette on either side. But this time, instead of having the full native Hephaestus element, he built a mini one, which is just this putative tyrosine recombinase, a zinc tolerance gene, and then a little bit of the flanking sequence. And then he transformed the fungus with this mini Hephaestus, again, to see if he would see it jumping out of the plasmid and into the genome. And that's exactly what he saw. So again, here you see all of these are now hygromycin resistant. So they had excision from the plasmid. And in all of these cases here are where it then made it into the genome. So you can see it was less efficient than sort of the native one, but it still does get back into the genome. And he's done some pooled sequencing and actually analyzed all those insertion sites to see that our other hypotheses about being target site specific and such also hold up. He then wanted to confirm that this really was through tyrosine recombinase mediated transfer. And so you can sort of just do an alignment. Cree recombinase is sort of the standard tyrosine recombinase that lots of people know and use. It's very well studied, so we know what the active residues should be. And so two that are important are this tyrosine here, and also this histidine here that, interestingly, in the original Captain in Kirk is still a histidine, but in many other starships, including in Hephaestus Captain, it's also a tyrosine. And so he mutated those to see what would happen. And you can see those results here that basically, if you do point mutations, then basically you lose, you stop seeing integration of the elements. So it no longer jumps into the genome. And interestingly, mutating in Hephaestus specifically, mutating that Y back to an H, which you can see over here, still seems to break it. So it's possible that these are actually working mechanistically in slightly different ways, which we also want to dig into a bit more in the future. So that's sort of, so if ICE is integrative and conjugative element, this really sort of proves that they're integrative. They are integrating into the genome and moving around in the genome. So then the next question is, are they conjugative? Can they be transferred? Sort of, do they encode the machinery to actively transfer themselves from one strain to an unrelated strain? And that's a much harder question to get at, but I'll sort of show some evidence that we do think that's what's going on. And the first example is here. So again, this is in Paecilomyces. This is where Hephaestus was, the species of fungus that Hephaestus was first discovered in. And they have multiple strains, and Hephaestus is polymorphic. It's in some and not others. And then you have this sister species, Paecilomyces formosus, which mostly doesn't have Hephaestus, except for one strain, which does have it. And it actually has what looks like another starship as well. And it's a bit of a question whether this was horizontally transferred, or you can invoke something like introgression. Maybe these two species aren't fully reproductively isolated, and they could just mate, and it could be transferred that way. So Andrew had a really clever way of addressing this, where he sort of took an agnostic approach and just chopped up the whole genome, and then looked at pairwise identity between all sort of synthetic parts of that genome. And so you get sort of a nice normal distribution, sitting somewhere around 80–85% when you do that. But then you get this big peak at basically 100%. And if you look at what parts of the genome are in there, it's exactly Hephaestus and this other starship. So if it was something like introgression, the rest of the background would have to have gotten sort of purged from the main genome, because it's really just these starship elements that still show that signal. And again, being almost no changes between these otherwise unrelated strains. Another example here that I'm pretty excited about is a horizontal transfer from something closely related to Fusarium, or maybe some of these insect-related fungi that's been transferred to Cryptococcus. And so, as I've been saying, the starships seem to be specific to Mycotina. This is a sort of subphylum within the Ascomycetes. Whereas Cryptococcus is a Basidiomycete yeast, so it's more closely related to mushrooms. It sort of should not have these elements as far as we can tell. And most Basidiomycetes indeed don't have this, and other closely related species of Cryptococcus don't have this. But this one species called Cryptococcus depoperatus, it turned out that it has picked up basically it now looks like a degraded starship, but it's had a horizontal gene transfer that brought over with it actually genes for arsenic resistance. So here's just a tree of an arsenic resistance gene. Cryptococcus should be over here with other Cryptococci, Cryptococcus depoperatus, but it's not, it's here. And if you do this for the other arsenic genes, because it's a cluster of actually 5 genes, they all show the same signal. Also of interest is this is specifically not Hephaestus. So Hephaestus actually comes out here. It's also got arsenic resistance genes. So this is totally unrelated element and an independent transfer, and a transfer that has crossed basically more than 500 years of evolution in order to do this. So even though the starships are sort of native to filamentous fungi, they're having impacts beyond that. And we can see shadows of this in Saccharomyces, so other Ascomycete yeast, as well as mushrooms, Basidiomycetes, things like this. So they're having bigger impacts outside of filamentous fungi even. And the more that we sort of dig into these elements and look at previously published cases of horizontal gene transfer, we can basically get them all down to being starships or looking like they are maybe degraded starships. So there's some really nice work showing big horizontal gene transfers in domesticated penicillium cheese that's used to make Roquefort cheese and Camembert cheese. Those are definitely starships. Tox-A is an important gene for multiple wheat pathogens that has been shown to be horizontally transferred between unrelated fungi. That's definitely a starship. There's virulence genes in the rice blast fungus. Those look like starships as well. So time and time again, actually, these starships are having really big signals of HGT in many different environments, in many different fungi. And so we don't think that it's accidental. We think that they are actually actively moving. Yeah, and I'll just sum that up here. So are they really conjugated? Do they really encode their own machinery to move? And so, as I mentioned before, we found that sort of auxiliary set of genes. And we think, we don't know what they're doing yet, but we think that somehow they're actually inducing hyphal fusion and potentially stabilizing the DNA intermediate to get to these starships transferred from one strain to another. But it is still possible that this is somewhat serendipitous, accidental. We know fungi do take up a lot of environmental DNA. So we'll have to do some work to really prove this. One sort of interesting case is actually Hephaestus, because it doesn't have any of those auxiliary genes anymore. So it's sad because we can't actually use that well-designed system to investigate this further. We have to now project another one. But then again, since we've seen Hephaestus move horizontally, does that mean that we're wrong? Or is the fact that we see it moving along with another starship mean that actually maybe as long as you have one active starship in a genome, it can actually mobilize multiple of these elements, which would have really interesting sort of intergenomic dynamics and intragenomic dynamics, potentially conflict among starships. There's a lot you can start thinking about there, which we're also really excited about. And just to dangle sort of an idea for anybody who's not working in fungi often, personally, I don't think this is just a fungal phenomenon. Fungi are the best set of eukaryotic genomes we have. We've been getting telomere to telomere assemblies of fungal genomes for a decade now, and at the population level for over 4 or 5 years. And we just discovered these things. If you look at other eukaryotes, we have no idea what's hiding in those genomes, right? We just got the first fully assembled human genome, last year. And so I think there might be big mobile elements hiding in other eukaryotes. And potentially one of them has already been found. And that's this thing called teratorn. So teratorn is a large mobile element. It's 200 kb, and it's spread across teleost fish. And it actually looks like it's some sort of fusion between a herpes virus type thing and a sort of regular DNA transposon called a piggyback. But it carries lots of other genes. If you look at the genes in there, you can't just ascribe them to sort of these two ancestral groups. And the papers also show a lot of diversity and claim that it could be a bunch of independent evolutions. But given how much diversity we see in the starships and things like this, it could also be one phenomenon, and that fish also have mechanisms for active HGT, just like fungi. And I think people are going to start finding these in many other systems. There's been some other cool work in Chlamydomonas showing similar things happening. So I think this is really just the beginning. And with that, I have a lot of people to thank. My lab, which is just my postdoc right now, Linnea, my former lab with Hannah Johannesson, and particularly Lore, who basically helped discover all of this. Emil is one of my major collaborators right now, who's started the Macrophomina work and really projected all of that and has been really great as well. And then, of course, Andrew, who did a lot of this studies in Hephaestus that I was talking about. And then various funding agencies, all from Sweden, for giving me money. And then again, for all of you for listening and the invitation as well again. Thank you.
Thank you very much, Aaron. Really fascinating findings. Are there questions from the audience? I have some questions if I'm allowed. It's really interesting that you find several copies of these elements in the genome. So the jumping process mediated by this recombinase, do they leave a copy of themselves behind? Because yeah,
no. So
how did you get ending with these several copies of these transposons in the genome?
So they look cut and paste.
Right.
And actually, other than Voyager, we have almost no cases where you have multiple functional looking copies. So of course, if you had one that sort of died and then recombination, you can get two in the same genome. And so we think that they're mostly single copy and that the multi-copy ones are more rare.
I see. And how frequent do they jump? Because this experiment is really impressive when you see that's not too many generations, right? So
yeah.
How frequently do you have any clue?
So in that experiment Andrew setup, it's about 1 in a million. So when he plates out 1 million spores, he would get one event of movement.
Okay, so, there is one question from Diego. Yeah, please go ahead.
Thank you. Could there be horizontal gene transfer inside Podospora itself? Because I remember when Podospora goes into senescence, there's mitochondrial genes living, coming out of mitochondria. Could they be also mediated by these starships?
So I don't think the starships are implicated in that. There are some really interesting plasmids that do mediate that. I think the PAL2 and PAL1 plasmids are known to mediate that. But that's actually where a lot of my ideas are coming from, of their ability to move between strains through hyphal fusion, because that's exactly how those mitochondrial plasmids move. But they, of course, don't initiate the fusion themselves. They're reliant on just sort of the rest of the gene to do that.
Thank you.
No problem.
Hi.
Yeah, Gabriel, yeah.
Nice, very interesting work.
Thanks.
I was wondering, so as you were commenting to Leonardo before, you usually have only one copy of these transposons or HGTs per genome. By that, what you will mean is that you will have at least 2 copies of the same genes that are carried by these chromosomes, by these transposons. So are these cases for gene duplications? And if so, then the question is, the genes are identical or they have diverged?
So yeah, so this is what we would call the cargo genes, so the extra genes that the starships carry. And those can have lots of origins. They can be duplicates of genes within the genome that they're currently in. Often they themselves look horizontally transferred. So there's many cases where, we try and build trees of these cargo genes and their closest relative is, again, in a different class of fungi. So there's a lot of mixing and matching and turnover of these cargo genes, and their evolutionary histories are very muddy. So it's not clear. The thing with the single copy in one genome that should be mentioned for anyone that's not really super familiar with fungi is they have this, many filamentous fungi have an efficient genome defense system, where if they have 2 identical copies of a gene, they'll actually start mutating them. It's called RIP. And so we can see signals of that actually sometimes, or we have dead copies, or we see that they've actually been targeted by this genome defense system. So that's also probably one of the reasons these don't really become multi-copy.
Okay, interesting. Thank you.
No problem.
There are some questions on YouTube. Let me read them for you. So the first one is by Fernando Hernandez. Do these starships may be a risk for phenotypic stability, thinking on the anthropogenic environment admixtures?
I'm not sure I completely understand the question, but I sort of think that these are actually helping provide, by being polymorphic in a population, they can help sort of encode different trade-offs very well. So things like arsenic resistance are actually a great example, because one of the ways to deal with arsenic is to actually pump it into the cell and then change its redox state and then pump it out. So it can actually be very bad if you're in a low arsenic environment to bring that into the cell. It can be worse than just sort of ignoring it. So you might not always want those genes around. So by having this polymorphic in the population and in different sort of heterogeneous environments where sort of some soils over in one area might be heavily contaminated and others are not, it allows you to have that flexibility and to gain these genes back after you lose them just by meeting a conspecific. I hope that addressed it.
Okay. Thank you. And there is a second one. What's the extent of freedom for the starships to move among species? What would be their effect on species with non-canonical genetic codes? or ambiguous ones?
So, yeah, so the first part is easier to answer. So we think that they're using sort of the standard host machinery for hyphal fusion. So fungi they have a very regulated system for determining whether or not they should fuse hyphae. Often it's a bad thing to do. You don't just want to fuse your hyphae with any old fungi out there because maybe they've got viruses, parasites, maybe they're actually somehow selfish and they could, take all your nutrients. So we think that they're co-opting these regulatory networks, and actually the genes that we see inside the starships support that, because these nod-like receptors, these NLRs, are very often used for this in fungi. And in normal cases, those are all mediated sort of by closely related species. So you'll go to the point of basically fusing your hyphae and then deciding, do we keep these fused or do we actually kill those cells and not fuse? If it's a closely related fungus. But if it's a very different fungus, you usually they do those interactions through more sort of pheromone signaling. So those colonies won't even grow close to each other. So they don't even try and fuse their hyphae. So that means that we would expect these to mostly move within sort of conspecifics and congenerics, and that transfers between more distantly related things would be more rare. For things with non-standard genetic codes, like the polymerase, all of that, like that should be the host machinery. So the only thing that the tyrosine recombinase says is induce a holiday junction to basically pop this thing out as a circular intermediate. So it would all be relying on the host machinery for it.
Okay, thank you. Okay, so do we have more questions? Okay, if not, thank you very much, Aaron, again, for your nice talk.
Thanks again.
Thank you very much. Really nice work. Thank you to everybody. See you next Friday.
Thank you very much.
Thanks. Thanks for
Thank you. Bye.